P.S. Buchanan.
So the talks today they huge range, so from pteropod ecology to trait-based modeling, and these talks are really just to either maybe introduce the results from a poster and entice you to come, or just introduce themselves and who they are as scientists, and to get you guys to interact with the students of those talks.
Yep, so I think we've got 18 speakers, and I'll have 3 minutes each, and I'll be counting. And so I'll stand up, for everyone who's speaking, I'll stand up when you get close, like 30 seconds. And I'll just be in the corner here. Okay, and I'll be diligent. So I think we're the actually first 2 speakers, so we'll set the bar.
It's alphabetical, but I didn't take my husband's last name.
Oh, and questions. We'll leave questions till the end, if anyone has burning questions. Otherwise, just approach someone.
And once you see a talk for the speakers, come right up so we get this rolling. So my name is Emily Brownlee. I'm a postdoc at the University of Maryland Center for Environmental Science, Horn Point Laboratory. And I'm just going to give you a little 3-minute blurb about my thesis research and what I'm doing now as a postdoc. So I worked here for my thesis research with Heidi Sosik, and I explored the seasonal patterns of ciliate communities on the New England shelf through integrative taxonomic analyses. I used an instrument called Imaging Flow Cytobot. It takes high-resolution in situ images of cells less than 200 microns that have chlorophyll fluorescence. So this includes mixotrophic and herbivorous ciliates. And these deployments have gone on since 2006 until recently. So we have these really high-resolution time series of these different ciliate morphotypes. And I was interested in taking a complicated time series like this and decomposing it into components that tell us something about seasonality. So an expected seasonal cell concentration, which is separate from interannual variability, which gives us an idea of the magnitude of that seasonality from year to year. Furthermore, with DNA sequencing on the rise and the fact that morphology does have its limits, I was interested in how genotyping can complement what we already know about seasonality that's derived from morphology. So a just quick teaser, I don't have enough time to really get into the results, but here's just one tintinid morphotype, and looking at the counts over these 3.5 years, I did some high throughput sequencing on discrete samples. And when I looked at the normalized OTU abundance, I found that it very well captured the seasonal cycle that we get from morphology. And while this didn't always hold up with all of these morphotypes, I saw that genotyping and morphology can really complement each other. And so please, if you're interested, come up and talk to me about it. So now I'm still interested in seasonal plankton dynamics, but I moved down the continental shelf and I'm working in the Chesapeake Bay, and I'm interested specifically in the wintertime. So in temperate coastal ecosystems, it's been thought that wintertime is a time of negligible productivity. But in spite of this, in the mesohaline regions of the Chesapeake Bay, we see that there's a peak in chlorophyll A in the wintertime. And in fact, this is the highest chlorophyll that we see all year. And most of it's actually happening below the picnic line, so in these deeper, darker waters. So I'm interested in exploring the fate and composition of this pool of POC and associated chlorophyll. And more specifically, I'm interested in the dinoflagellates that dominate the wintertime blooms. And so dinoflagellates have been thought to be mixotrophic, and this allows them to have a competitive advantage when light is low and photosynthesis is constrained. And so now I'm using, in the lab, a dinoflagellate Heterocapsa rotundata, which has been proven to be mixotrophic under limiting light conditions. And I'm trying to develop tools to quantify mixotrophy. So using natural stable isotope abundances in the lab with the organism, providing it an array of conditions of light that induce mixotrophy. So with that, I will pass it on.
Okay, well, thank you.
Yeah.
Okay, so for a total change in scales, well, first, I'm Piers. I'm from Tasmania. I'm currently at Princeton until the end of the year on a Fulbright scholarship. And during my PhD, I was interested in global changes in the carbon cycle over thousands of years. So millennial scales. Millennial scales, interglacial, glacial cycles, that sort of thing. now actually going more into deep time, and I'll show you that in a moment. One of the things that I was really interested in during my PhD was building a marine nitrogen cycle within a global ocean model. And during this process, I got particularly interested in the nitrogen fixes. And this was for 4 main reasons. 1, they fix nitrogen, so that's pretty obvious. 2, they efficiently consume all phosphorus in the environment, inorganic, non-organic. They're really good at doing that, drawing it in. 3, they have really high C to P ratios of the organic matter, so carbon-enriched organic matter, which you can see by the yellow arrows. And then 4, they're really sensitive to aeolian-borne sources of iron in the modern, at least in the modern climate. So when I kind of try and pair the nitrogen cycle and nitrogen fixes with glacial and glacial cycles, we know that these glacial and glacial cycles had large changes in the carbon cycle and atmospheric CO2 of roughly 100 ppm. They also had really extreme changes in how much dust was in the atmosphere and how much the ocean was fertilised by iron. And so I thought we could probably quantify the role of nitrogen fixes in the carbon cycle over these glacial and glacial cycles. And so I did all these idealised experiments where we test different representations of nitrogen fixes, different different depositions of aeolian iron to the ocean's surface, and also mixing up the ocean, excuse the pun, but changing the different circulations. And this linear relationship kind of pops out where for every increase in nitrogen fixation globally by 1 teragram of added nitrogen per year, roughly 0.5 ppm of CO2 is drawn down in the ocean and held there. So let's say for a 20 teragram increase in nitrogen fixation per year, you might have 10 ppm that's actually drawn into the ocean and stays there. And so nitrogen fixation pops out as this non-negligible ingredient into the glacial and interglacial cycles. And so that was for my PhD, and then while I'm here at Princeton, I'm working with a group to do some deep time stuff. And so what I'm showing you is a grid that has been implemented within the model. So it's a paleobathymetry from 55 million years ago during the Paleocene, and we're reconstructing the nitrogen cycle at that time and modelling nitrogen isotopes. So there were some massive changes between 60 to 30 million years ago in the nitrogen cycle as the climate cooled down and ice sheets developed over Antarctica. And I'm just about out of time, so I'll stop there. Thanks.